Hello everyone, this is Sapna from RPIIT Medical and Technical Campus. In previous class, we have discussed glycolysis. In today's class, we are going to discuss about gluconeogenesis, uh, which is the another one sequence of carbohydrates metabolism. So here, gluco means glucose and neo means new. Genesis means formation. Here is the synthesis of glucose from non-carbohydrate sucrose, and the site for this synthesis is mostly takes place in liver, approx 1 kg of glucose per day, and in kidney also. Gluconeogenesis occurs mainly in cytosol. Some precursors are produced in mitochondria. Notable precursors are pyruvate, glycerol, lactate, propanate, glucogenic amino acids. So why do we synthesize glucose? We synthesize glucose because the need to maintain glucose level within a narrow range in blood. Brain, erythrocytes, testes, and kidney medulla are depend on glucose for continuous supply of energy. Skeletal muscles in excretion anaerobic conditions use glucose at a rapid rate. In fasting to meet the basal requirement of body for glucose essential for survival effectively prevents the accumulation of certain metabolites in blood, for example, lactate, glycerol, propanate, etc. And here is the reaction for gluconeogenesis. In gluconeogenesis, there is chorus cycle which starts from lactate and pyruvate. Pyruvate is converted into oxaloacetate, and here ADP is converted into ATP. Then oxaloacetate is converted into phosphoenol pyruvate in presence of PEP, phosphoenol pyruvate kinase enzyme. And here is the carboxylic kinase enzyme is also present. And then the phosphoenol pyruvate in presence of enolase enzyme is converted into 2-phosphoglycerate in presence of phosphoglycerate mutase enzyme, and 2-phosphoglycerate is converted into 3-phosphoglycerate in phosphoglycerate mutase and in presence of phosphoglycerate mutase enzyme, and then 3-phosphoglycerate is converted into 1,3 bisphosphoglycerate in presence of phosphoglycerate kinase, and ATP is converted into ADP, and here 1,3 bisphosphoglycerate in presence of glycerate 3-phosphate enzyme is converted into DHAP, that is glycerol, and glyceraldehyde 3-phosphate, and then glyceraldehyde 3-phosphate is in presence of aldolase enzyme is converted into fructose 1,6 bisphosphate, and fructose 1,6 bisphosphate in presence of fructose 1,6 bisphosphatase enzyme is converted into fructose 6-phosphate, and fructose 6-phosphate is converted into 6-phosphate, and then glucose 6-phosphate is converted into glucose in presence of enzyme glucose 6-phosphate, and in chorus cycle the location will enters in liver. Then what will happens in liver? From muscles to perform gluconeogenesis in liver, gluconeogenesis takes place, and here we can see the entrance of glucose from muscles to liver. Here we can see in liver there is glucose, and glucose is supplied to muscles, then it produces 2 ATP and converted into 2 pyruvate, and then it is in reversible form to 2 pyruvate in reversible form converted into 2 lactate, and these 2 lactates goes to liver, and these 2 lactate also in reversible form uh, they are converted into 2 pyruvate, and 2 pyruvate gives 6 ATPs and converted into glucose in liver, and it is transported through blood. Here are some names of glucogenic amino acids from which glucose is formed. These amino acids are arginine, histidine, thyrosinine, tyrosine, aspartate, cytosine, glutamate, glutamine, pyrroline, vanillin, asparagine, alanine, glycine, isoleucine, and phenylalanine. These all are the amino acids which performs gluconeogenesis. Thank you.